Hello, everyone. My name is Jack Webb, and I'm a first-year master's candidate in the Department of Anatomy and Cell Biology, and I'm supervised by Dr. Trevor Shepard. And I'm really excited to share some of my research project with you, which is titled ULK1 is required for autophagy in ovarian cancer spheroids. So epithelial ovarian cancer, or EOC, is the most lethal gynecologic cancer due to its late-stage diagnosis and no effective treatment strategies for treating chemo-resistant disease. And so EOC is unique as metastasis occurs via dissemination of cells directly into the peritoneal cavity. These cells then aggregate and survive in the ascites fluid in the peritoneal cavity as spheroids. And so our lab has shown that spheroids undergo numerous phenotypic changes, such as inducing autophagy. And so macroautophagy or autophagy is a highly conserved intracellular degradation system that maintains cell homeostasis when activated by cellular stress. And so in response, autophagy facilitates the turnover of damaged organelles, removing damaged or misfolded proteins and harmful components while replenishing essential biomolecules required to fuel biosynthetic pathways and metabolic processes. And so cancer cells can hijack autophagy to overcome nutrient deprivation, hypoxia, and can also confer chemo resistance. So a key regulator of autophagy is ULK1, or UNC51-like autophagy activating kinase 1. And this is a serine-threonine specific protein kinase that senses upstream signals from nutrient sensors, such as AMPK and mTOR, which can lead to the activation or prevention of the downstream autophagy pathway. And so the rationale for my project is that high-grade serous ovarian cancer spheroids have increased ULK1 protein expression that parallels autophagy activation, and that ULK1 knockdown and inhibition blocked autophagy activation and reduced cell viability. Therefore, we hypothesize that inhibiting ULK1 will reduce EOC tumor growth and metastasis by targeting its crucial function in autophagy-mediated cell survival. The two objectives for my project are as follows. The first is to evaluate the effect of CRISPR-mediated ULK1 ablation in experimental models of EOC metastasis. And the second is to assess ULK1 requirement for EOC tumor establishment and metastasis in xenografts. So the first step in characterizing these particular ovarian ULK1 knockout clones was to perform molecular analysis by means of Western blot that investigated key proteins that are involved in the autophagic pathway in order to establish autophagy status in these cells. And so here in panel A is a representative Western blot of ovarian, parental, and ULK1 knockout clones investigating those proteins. And so the first is ULK1, and the second is P62, which is an autophagy substrate. And so increased levels of P62 can be observed when autophagy is impaired. The next proteins are LC31 and LC32. So LC31 is processed into LC32, where LC32 is an autophagosomal membrane marker. And so the quantification of the LC32-to-1 ratio can help indicate if autophagy activation is impaired. And so a decreased ratio, or a ratio close to 1, is observed when autophagy is impaired. And so when we look at these two proteins together, we can get a simple understanding how ULK1 status can alter autophagy. So here in panel B are the quantifications of these proteins. And we see here there is an increase in ULK1 in spheroids, where there is ULK1 loss in these ULK1 knockout clones. Looking at P62, the levels of P62 are different, but overall, most of these clones exhibit increased levels of P62, seen here in clone 1, clone 15, and clone 5. And looking at the LC32-to-1 ratio, the trends are a little bit more obvious, with all clones exhibiting a decreased LC32-to-1 ratio, and most are around 1, which indicates limited conversion of LC31 to LC32 and decreased autophagy activation. It was then critical to assess spheroid formation in each of these clones, since we know that spheroids offer EOC cells with increased viability and also protects them from chemotherapeutic insult. So what I have depicted here in panel A are EOC cells grown in spheroid condition and imaged at day 3, 7, and 10. And we see that there are clear morphological differences in terms and qualitative differences in terms of spheroid shape, density, and number and size. And they also appear to have reduced spheroid integrity compared to the parental. So we then wanted to delve into some quantitative approaches, such as assessing viability and apoptosis in these spheroids. So to measure the viability of these spheroids, we used a trypan blue exclusion assay. And this graph here shows in panel B the relative viability of these ULK1 knockout clones compared to the parental at the specified time points at day 3, day 7, and day 10. And so overall, there is definite variability in the viability amongst these ULK1 knockout clones. We see that clone 1 here in red has increased viability at day 3, followed by decreased viability at day 7 and 10. Clone 13 has decreased viability across all time points compared to the parental, where clone 15, 14, and clone 5 all exhibit increased viability compared to the parental across all time points. So we further assessed viability using a cell titer glow at day 3, and cell titer glow estimates the number of viable cells in suspension based on the quantitation of ATP. And so we see using cell titer glow that there are more viable cells in all clones compared to the parental. We also want to determine whether or not there were changes or a decrease in apoptosis that resulted in more viable cells. So we therefore performed a caspase glow assay, which measures caspase 3 and 7 activities, which are activated during apoptosis. And we see that there are increases in apoptosis in all ULK1 knockout clones compared to the parental, with at least a 50% increase in caspase activity in these ULK1 knockout clones. So shifting gears from our spheroid model system here in panel E, we wanted to assess each of these clones' sensitivity to carboplatin in adherent condition using an Almar blue assay. And surprisingly, we see that these ULK1 knockout clones have decreased sensitivity to carboplatin, reflective of the higher IC50 values shown here in the graph. These results can be partially explained by this growth data here shown in panel F, where these clones grow significantly slower than the parental, reflective of the higher doubling time compared to the parental here. And so lastly, in panel G, these off-carrate spheroids were treated with their adherent carboplatin IC50 value, and their sensitivity to carboplatin was then assessed using an Almar blue assay. And so these bars, the bars on the left represent no treatment, and bars on the right represent carboplatin treatment. And surprisingly, we see that these ULK1 knockout clones appear to be resistant to carboplatin, indicated by the increase in fluorescence in the treatment groups. So these next experiments were performed in FT190 cells, which are an immortalized human fallopian tube secretory epithelial cell, and these cells do not form tumors. And the fallopian tube is the site of origin for most high-grade serous ovarian cancers, but not the other histotypes. And so these cells are used as a control. So here in panel A is a representative Western blot of the autophagy markers that I had previously talked about. And so in panel B are the quantifications of those autophagy markers, so LC3 2:1 ratio, as well as P6P2. And looking at the LC3 2:1 ratio, we see that LC3 processing appears to be intact, with ratios all comparable to the parental and at least a ratio above 2. And looking at P6P2, we see that ULK1 knockout clones exhibit decreased levels compared to the parental, and this further indicates functional autophagy activation. We also performed a trypan blue viability assay on FT190 spheroids at day 3, which are shown here on the left in panel C. And so there appears to be increased smaller spheroids or more cell debris in these ULK1 knockout spheroids. And so we performed a trypan blue viability assay to determine viability, and this is shown in panel D. And the trypan blue viability assay indicates that there are fewer viable cells in the ULK1 knockout spheroids, indicated here in black, and that there is an increase in the number of non-viable cells shown in red. And this is further emphasized with a caspase glow assay here in panel F, or in panel E, sorry, that has been normalized to cell titer glow. And we can see that all these clones exhibit increased apoptosis compared to the parental. So these results have some important implications. So ULK1 loss impairs autophagy activation in OVCAR-8 spheroids, but it's not impaired in FT190 ULK1 knockout cells. And so this suggests that there is a reliance of ULK1 for autophagy activation in cancer cells specifically. And so this highlights that the therapeutic potential of targeting ULK1. And I've shown that there are a subset of clones that trend away from a hypothesis, and therefore we have the potential to uncover some resistance mechanisms resulting from autophagy inhibition. And so moving forward with my project, I will be generating dose response curves and IC50 values in OVCAR-8 ULK1 knockout spheroids, and this will solidify sensitivity to carboplatin. I will also be generating HAY8 ULK1 knockout cell lines, which will allow us to generalize our results in identifying ULK1 as a novel therapeutic target in high-grade serous ovarian cancer. And I will also be generating luciferase expressing clones used for xenografting and BLI measurements. And I'd like to thank Dr. Shepard and my lab members for their continued support. And I'd like to thank the Department of Obstetrics and Gynecology for their funding, as well as our additional funding sources. Thank you for listening.